Well, good morning, good afternoon, depending upon your geographic location. Thanks for joining us for another one of our series in the drug-drug interaction clinical decision support series sponsored by myself at the University of Utah and on behalf of my colleagues at the University of Arizona, University of Pittsburgh, and the University of Washington. Thanks for taking the time to join our webinar today. Today, I'm really excited that we have a presentation by Dr. Phil Hanston, Professor Emeritus from the University of Washington. Many of you know Phil, and he really needs no introduction in the drug-drug interaction space. He is probably one of the world's preeminent scholars in the area of drug-drug interactions and has been publishing on drug-drug interactions since I was probably in diapers. Anyway, no, Phil is a wealth of knowledge, and today he's going to share some of his insights as it relates to tamoxifen drug-drug interactions. We're really excited about this presentation. I think those of you in attendance and those of you that might be watching this later, because we will record and post this to our website at bdi-cds.org, our website, we'll find that this is a very intellectually stimulating and also, to some extent, eye-opening presentation from Dr. Hanston. So without any further ado, I'll turn it over to you, Phil. Thank you very much for joining us today.
Great. Thank you very much, Dan, for that nice introduction, except for the thing about the diapers. I'm going to get back at you for that at some point. I'm old, but I'm not that old. Okay. Okay, I have a very simple goal for this webinar, and that is by the end of this lecture, I hope to propose a few simple steps to reduce the risk of tamoxifen drug interactions. And in the process of the lecture, I want to justify the recommendations that I'm going to make at the end. So I hope you will agree with them by the time I'm done. Controversy. This is one of the most contentious drug interactions in history, and I have been doing it a long time, not since Dan was in diapers, but for over half a century. And I think it gets first place as far as how controversial it is compared to others. So is the problem too many cooks? We certainly have a lot of cooks. We have oncologists and other clinicians. We have statisticians, epidemiologists, geneticists. We have people like myself in drug-drug interactions and kinetics. And actually, I look a lot like that guy on the right. But the problem is that the cooks sometimes disagree on tamoxifen drug interactions. Now, we can't get rid of any of the cooks to solve the problem. We need all these cooks. So it's not a matter that we can just say, well, let's get rid of some of them and then we won't have so much controversy. I haven't seen any strangulations yet, but it has gotten pretty heated. So perhaps injudiciously, I'm going to recommend we add another cook, philosophy, although I think that this cook can maybe help resolve some of the disagreements among the others. And we'll get to that in the second half of the lecture. There are a couple of areas of agreement, surprisingly, and one of them is that tamoxifen metabolism and distribution is very complicated. Here's one depiction of that. It's pretty complex, and it's not just drug metabolism, but also transporters are involved. So I think we could safely say, I'm going to give tamoxifen first place in the pantheon of drug metabolism and distribution as far as complexity is concerned. Now, I have simplified it on this slide, and I included just the pathways that I think are the most important from a drug interaction standpoint. So I used Occam's razor to do this. William of Occam, as you may recall, recommended that we not make things more complicated than necessary. So he would probably not like this and would vastly prefer this. Another area of agreement, actually, is that tamoxifen is a prodrug and endoxifen is the active metabolite. There's general agreement on this as well. On both sides of the argument, actually, people agree on this. So, but from there on, we get into the disagreements, the main one being, does decreased 2D6 activity from genes or drugs reduce tamoxifen efficacy? And it makes sense that this would come up because the pathway between tamoxifen and endoxifen goes through, the road goes through 2D6, no matter how you, which pathway you take, you've got to go through 2D6. So it makes sense that lowered 2D6 activity from genomics would be a problem. And there are several studies suggesting exactly that. Here's cancer recurrence in EMs on the left bar, people with normal 2D6, IMs, intermediate, and PMs on the right, people with low 2D6, and a nice stepwise increase depending on the activity of 2D6 in these people. And as I said, other studies have confirmed this, and yet still other studies have failed to confirm this. So this one's controversial still. Also controversial is the effect of 2D6 inhibitors. And it's the same issue. You've got to go through 2D6 to get to the active metabolite of endoxifen. And it's clear that if you give 2D6 inhibitors that you do reduce endoxifen concentration. This is actually not controversial. Control bar on the left and people on paroxetine on the right, a very large decrease. In EMs, it was 64% in the active metabolite. Also, if you switch from paroxetine, a potent 2D6 inhibitor, to escitalopram, which has very little effect on 2D6, as you can see, there's a dramatic increase in endoxifen AUCs. So this one, the effect of 2D6 inhibitors on endoxifen levels is pretty well established. Now, the question, though, that is more debatable and more controversial is if you have people on these SSRIs, for example, that inhibit 2D6, does that increase breast cancer recurrence and mortality? This study done by Dave Yerlink's group up in Toronto, looking at paroxetine, as you increase the overlap of paroxetine with tamoxifen, there was a stepwise increase in breast cancer mortality. And again, other studies agreed with this and other studies did not find this. So this one is controversial as well. And the controversy goes up to today. Here are two quotes from 2022 on opposite sides of this. The top quote, 2D6 gene polymorphism has a profound impact on the effect of tamoxifen. The bottom quote saying precisely the opposite, that you don't have to worry about antidepressants and tamoxifen. So we still have these diametrically opposed views on the issue of 2D6 and tamoxifen. So the controversy continues. People disagreeing on the effect of 2D6 on tamoxifen efficacy. So I'd like to try to address why it is. Why is it that we have so much difficulty in sorting this out? Probably more money has been spent, and here's another first for tamoxifen. I would wager that more money has been spent on tamoxifen drug interaction studies than any other drug interaction in history. So you would think that with all of those people working on it and all that money, that we wouldn't have all this disagreement. Well, I think a big part of the reason is what I'm going to talk about next, and that is the pitfalls in the, or the limitations in the published studies. Now, these studies are usually observational. And as you know, observational studies are fraught. They're difficult. It's difficult to sort out causal relationships or lack of causal relationships. And Tyler Vigen shows this with the number of people who drowned by falling into a pool correlates with films Nicolas Cage appeared in. And he also found that the number of people who died by becoming tangled in their bed sheets correlates with per capita cheese consumption. So you do have to be careful about making associations or lack of associations in these observational studies. So there are some general limitations, and we're not going to go into any detail on these. We're going to focus on the drug interaction part of it, but there are some general limitations of tamoxifen studies. Adherence is very difficult to assess in these large observational epidemiological studies. Especially in the past, there was inadequate genomics done. This is improving. Inadequate study size is still a problem. Sometimes they just don't use enough people. Different endpoints are used, either breast cancer mortality or recurrence and various other things. This one is important. Menopausal status is often ignored. And there is actually some agreement on both sides of the argument that premenopausal patients are probably more at risk from these interactions than postmenopausal patients. Yet many studies just throw them all together in one big pot, and the results you get may or may not be useful to one group or another. wrong way, sorry. Inadequate study duration is still a problem. You have to study this for several years, and some of the studies that involve only a couple of years it's just not long enough to look at this. And finally, variability in how they define concurrent use of tamoxifen with the other drugs. So there's lots of general limitations that we're not going to talk about anymore, but there are a lot of headwinds right from the start in doing these studies. But what we're going to focus on today are the specific flaws in the tamoxifen 2D6 inhibitor studies. These are mainly failing to control for the effects of other drugs or genes on the enzymes or transporters. One good example is failure to control for other 2D6 inhibitors. So let's say you're studying SSRIs and tamoxifen, but you failed to look at other 2D6 inhibitors correctly. Some didn't control at all for the other 2D6 inhibitors, and others used flawed lists, as shown here. And I'm indicating most negative studies used flawed list of 2D6 inhibitors, if they use them at all. And that first group, that in two widely cited negative studies, they're always cited when people are trying to claim this interaction doesn't exist. 2/3 of the drugs they controlled for by listing them as 2D6 inhibitors were not 2D6 inhibitors. In the bottom there, fewer than half of the drugs listed as 2D6 inhibitors actually were inhibitors. That study was also too short, and the endpoint was total mortality rather than breast cancer mortality. So, and I could give you slide after slide showing flawed lists of 2D6 inhibitors. It's a huge problem. But this, I'm going to recommend, if there's one key takeaway point from this lecture, this is it. That enzyme inducers have been largely ignored in the studies and largely ignored pretty much with tamoxifen in general. And they are probably more dangerous to patients on tamoxifen than patients on 2D6 inhibitors. And if you combine the two together, if you have somebody on a 2D6 inhibitor and an enzyme inducer at the same time, I think it would be very difficult for them to respond at all to tamoxifen. Why do I say that? Well, enzyme inducers affect all of the pathways you see in red, and all of these things tend to reduce dramatically, as I'll show you in the next few slides, dramatically reduce serum endoxifen. So the active metabolite serum concentrations are dramatically reduced, probably more even than with 2D6 inhibitors. And it's a double whammy. If that weren't enough, then look at the cancer cell over on the right. The efflux out of that cancer cell, which you don't want, you want it to stay in the cancer cell. That efflux is almost certainly increased by enzyme inducers, both by ABCB1, peak glycoprotein, as well as ABCC2. So it's a double whammy with lowered endoxifen and increased efflux. I would suggest that people on inducers probably are very unlikely to respond to tamoxifen. And sorry about that thing that just dropped down. It'll go away in a second. Okay, tamoxifen and aminoglutathione. Aminoglutathione is a potent enzyme inducer. At least I think it'll go away. And you can see here, under control circumstances versus people on aminoglutathione, a 93% reduction in endoxifen plasma concentrations. So a major reduction and probably subtherapeutic. And 3 patients had 0 endoxifen concentrations. And I'm not sure how to get rid of that thing at the top. I don't know what I did to cause it, but are you seeing that thing that's obliterating part of the?
No, Phil, we're not. And that just might be part of the Q&A that is coming through. So
okay, great.
We'll answer the questions toward the end of the presentation. We've reserved some time for that today. So hopefully people can stick around until the end or toward the end, and we'll answer those questions.
Perfect. Okay, great. Well, it's not hindering me, but it came down on mine. So, okay, so 3 patients had 0 endoxifen concentrations, which now there's some debate about what the therapeutic level of endoxifen should be, but 0, I think most people would agree is not enough. Rifampin does the same thing. It produces dramatic reductions in endoxifen plasma concentration. And this is a busy slide, so bear with me for a second. The main thing is that little box, that red box in the middle there, because those are people on rifampin And the left one is rifampin in GNM, which is normal 2D6. The middle one is GIM, intermediate metabolism of 2D6. And the right one is GPMs, which are poor metabolizers. It didn't matter. 100% of patients on rifampin had subtherapeutic endoxifen concentrations, regardless of the 2D6 genotype. So it would appear that enzyme inducers are a serious problem. Just a little bit of more information. Here's a patient on phenytoin who was started on tamoxifen. The patient is on the bar on the left, and her endoxifen concentrations were less than half even of PMs, people with no 2D6, and way less than EMs, people with normal 2D6. So I think that the evidence is pretty conclusive that enzyme inducers are bad news in people on tamoxifen, yet none of the studies, not a single study, controlled for enzyme inducers. And the inducer they should have controlled for and didn't is this one. This is a huge problem for these studies. Tamoxifen and St. John's wort. St. John's wort is known to be a inducer of all sorts of enzymes and also the ABCB1, all of which have the potential for affecting tamoxifen. And here's the problem. Tamoxifen produces menopausal symptoms. This is not debatable. Everybody knows that menopausal symptoms are much more frequent in people and often severe in people taking tamoxifen. And the problem with this is that St. John's wort is widely recommended in the treatment of menopausal symptoms. If you go online or in other sources, you see frequent recommendations that St. John's wort is useful in the treatment of menopausal symptoms. So the difficulty here is that studies of 2D6 inhibitors didn't control for the use of St. John's wort, which could have been used instead of SSRIs like paroxetine. So the SSRIs are often used to treat menopausal symptoms. St. John's wort is used to treat menopausal symptoms. So what that would mean is then that this would mask an effect of the SSRI because the control group would also have reduced efficacy. And this is especially problematic because St. John's wort is probably more potent than the SSRIs in reducing the effect of tamoxifen. So this is a major problem with the studies that have been done so far because this confounding factor could easily have been used in place of the SSRIs. They also failed to correct for ABCB1, which is P-glycoprotein, is AKA P-glycoprotein. I'm going to use ABCB1 in this presentation, but it's the same thing. And here you would expect that if you have increased ABCB1 activity, that you would increase the efflux and reduce the efficacy of tamoxifen. And sure enough, here's a study looking at people with high, medium, and low ABCB1 activity. And as the ABCB1 activity declines from A to B to C, recurrence and metastasis goes down. And these are all people on tamoxifen. So in this case, you do want low level or low activity of ABCB1 to maximize the effect of the tamoxifen. And what happens if you combine low 2D6 with high ABCB1? Well, this was pretty dramatic. The bar A on the left are people with intermediate metabolism for 2D6, so lower 2D6 and higher ABCB1. And for them, it was only 12 months before there was recurrence on average of recurrence or metastasis, whereas either one of those problems alone, it was 48 months. So a rather dramatic change in how long it took for them to have a recurrence, depending on the 2D6 combined with the ABCB1. And again, the studies didn't, other than this one study that looked at this, all of the other observational studies failed to look at this issue of the ABCB1. And there are various ways you can get there. The one we talked about just now was the one in purple, genomics affecting the ABCB1 and the 2D6, but it could also be drugs. In the red in the lower right, the red is two drugs, one an inducer, one an inhibitor. And then there could also be hybrids where one's an inducer or an inhibitor and the other is genomic. So there are a lot of ways you could get there, again, completely ignored. And probably any of these situations would increase the risk of cancer recurrence in these people. A different study looking at ABCB1 and 2D6, first they did find that 2D6 PMs, people with low 2D6, did have worse outcomes than EMs. But look at the effect with ABCB1. In that Kaplan-Meier curve on the bottom, survival, low ABCB1, which is what you want because survival was pretty good there, intermediate in the middle, and look at the Kaplan-Meier curve for high ABCB1 activity, rather dramatic drop off in survival in those people. Again, the studies, other than these two that I've just talked about, pretty much ignored that. So what about, this brings up another question that some of you have probably already been thinking about. Would P-glycoprotein or ABCB1 inhibitors counteract the effect of 2D6 inhibitors? So if you have a 2D6 inhibitor that maybe inhibits tamoxifen, but then you have, and there are probably 40 or 50 drugs that are P-glycoprotein, ABCB1 inhibitors. None of the studies controlled for this, and yet they could perhaps, and we don't know for sure because it hasn't been studied, but theoretically, at least, you would expect those inhibitors to counteract the effect of the 2D6 inhibitors. So this is a question mark, but certainly it's plausible from a biological standpoint. I had a slide with all 40 or 50 ABCB1 inhibitors on it, all drugs that inhibit that, and I accidentally deleted it this morning, so you don't have to look at that slide. Another transporter that you've already seen on the slides, ABCC2, is probably very similar, where you have higher levels of this reducing efficacy of tamoxifen. And again, you've seen this before, increased efflux. And they found similar results with this transporter, cancer recurrence stepwise going down as ABCC2 activity goes down. And this is a very scary slide. Probably the scariest slide I'm going to show you, maybe even scarier than the enzyme inducer stuff that I talked about a moment ago. In people with the highest let me start with the 2D6. People with the lowest 2D6 and the highest ABCC2 had a catastrophic 45-fold increase in the recurrence of breast cancer compared to people that one was by definition people with the lowest number of risk factors. That's a bit scary, and we need to have more information about this. And the fact that the studies didn't even look at ABCC2 is critical. So just to summarize, lots of things can affect a lot of drug interaction factors, and genomic factors can theoretically or in fact known to affect tamoxifen outcomes. The ones on the left with the UGT and the SULT, I didn't even talk about those because we don't have time, but they probably also can affect tamoxifen outcomes and may be altered by genomics. So it's a big problem. It's complex, as you can see. Maybe not as complex as this toilet I ran across on a trip a couple decades ago, but this is the lid. Warnings and cautions on your toilet seat. I don't know if we're advancing as a society when I have to read a whole bunch of caution. I was afraid to sit down on this thing. But anyway, so the question comes up then, given all of these problems with the studies, the published studies, were these negative studies or were they failed studies? Professor Walkup was talking about a different issue, adolescent depression, but he said many trials, quote, are failed trials because of substantial methodological problems rather than negative trials. So the question I raise here is all of the negative trials and the positive trials also had flaws as well, but are all of those negative trials and people claiming that this interaction does not exist, are those based on negative trials or failed trials? So the problem, I want to assure everybody, the problem is not the competence of the researchers doing these studies. It's the difficulty of the task. It may not rise to the level of Sisyphean, but it definitely is a very difficult group of drug interactions to study. There's this unique complexity of tamoxifen metabolism and transport. Dozens, actually, when you throw in the ABCB1 inhibitors, it's probably getting closer to 70 or 80 drugs that can affect tamoxifen disposition or effect. There are many genomic influences, as we've discussed already, many of which are not considered in the studies. the studies have to last a long time, several years at least, before you can really determine whether there's an interaction. The outcome is elusive because you're looking at prescription failure, or you're looking at therapeutic failure, rather than with many drug interactions, we're looking at adverse outcomes, bleeding in somebody on warfarin, or serotonin syndrome, or some kind of, or drug toxicity, instead of something that's a little bit more elusive, such as therapeutic failure. You need large study sizes, and people often don't achieve those difficult. We already talked about this. What does concurrent use mean? Some people consider just 6 months of concurrent use is enough to count. Other people use other definitions. And as we've talked about, effective menopausal status, that really shouldn't be ignored and often is. So David Yerlink from Toronto states, for all of these reasons, the reasons I mentioned on the previous slide, the tamoxifen SSRI interaction is perhaps the most difficult drug interaction to explore using the techniques of pharmacoepidemiology. And I think we need to appreciate this, that this is a very difficult interaction to deal with. So now we're to the philosophical part, which I think could help solve some of these problems. So I've created a lot of difficulties here in saying that we really can't trust a lot of the studies that have been done, and there's a lot of differences in the outcomes, et cetera. But I think that philosophy may ride to the rescue here and help us sort out how we should handle these things. Given the uncertainty that's clearly there, how do we handle this uncertainty? And the philosophy I'm talking about is not the meaning of life kind of stuff. This is what happens when your dog eats your philosophy homework. He's contemplating the meaning of life. We're not talking about that kind of philosophy, although that's fun to talk about and interesting. We're talking about more of the hard-nosed stuff, like William James, who was a physician and a philosopher. And when somebody asked him, what is philosophy? He said, a peculiarly stubborn effort to think clearly. So this is the kind of philosophy that I'm going to try to apply to the tamoxifen stuff. So the problem as I see it is that this hypothesis that tamoxifen efficacy is reduced by 2D6 inhibitors, we've taken a one-dimensional approach. We have believers who say, yes, it does, and there's still plenty of people in that category. But we also have a lot of naysayers who are saying that it's unlikely or even clearly not true. My personal position is in between, in the possible area there, but even if you call it possible, there's a lot we can do with that. So one-dimensional approach, and you can see I have room for 3 here, and this is how most of the studies have looked at this, and most of the researchers have looked at just this one thing. What is the probability that the drug interaction is real? And what we really need to do is take step 2 and 3. Step 2 is the severity of the potential drug interaction. These have to be linked together, not just considered separately, but linked so that you can make a better decision. Now, a philosopher of science, Richard Rudner, said, basically the question he's asking is, what if you're wrong? And he said in his paper, before we can accept or reject any hypothesis, the value decision must be made in the light of the seriousness of a mistake, that the probability is high enough or the evidence is strong enough to warrant its acceptance. And here, of course, he's talking about tentative acceptance. So the one-dimensional approach is just to look at whether the probability of whether it's the interaction is real or not. The Rudner approach is to add in another, the vertical axis here, the severity of the potential adverse outcome. And so I've put this into a form that I think is easy to understand with the threshold for action, that diagonal red line there. If you're in the lower left of that, no action needed. If you're in the upper right, action is needed. So for some interactions, you don't need philosophy for some interactions. Colchicine and clarithromycin, very well established, potentially fatal. You don't need anybody to ponder whether this is something you need to do something about. Obviously you do, and usually it's not give the 2 together. You also don't need philosophy to decide whether triptans and SSRIs are okay to give together, despite the labeling, which refuses to change. The evidence that this interaction is real is very low. In fact, the evidence clearly suggests that it doesn't exist. So you don't need help with that one either. Where we have the problems are in that middle group, the possible and probable interactions, especially the possible ones, to decide whether action is needed or not. Now, the tamoxifen 2D6 for believers, people who think this is real, it's pretty clear. If it's probable, on the probability scale, the severity is pretty severe, breast cancer recurrence or metastasis or even a fatality. Yes, they would recommend taking action. But the problem is that even if you're one of the naysayers and you say it's unlikely, when you consider the severity, when you link that unlikely probability to severity, you still get, at least in the way I've drawn it here, into the action is needed category. So I think the failure of looking at the severity and linking it with the probability is part of the problem. Now, I would put it here. My personal view is that this interaction is possible, maybe at the high end of possible, but and given the severity, my view is that if it's possible, clearly we should take action. Now, that's the two-step, but we actually need to add a third step, and we need the help of Blaise Pascal, one of the most incredible intellects in human history, in my view. He was a mathematical genius, co-inventor of probability theory. He was a scientist and inventor in hydraulics and other things. And for our purposes today, he had profound insights into the human condition. And if I didn't feel insecure enough, he was also an incredible humanitarian. He spent a lot of time helping the poor, et cetera, et cetera. So Pascal would recommend that we use a three-step process. So he would take what Rudner said, step 1 and step 2, but Rudner stopped there, and Pascal would add a step 3. So Rudner didn't even mention Pascal, even though Pascal had a better explanation of what he was proposing. But this is not unusual. It follows what I call Gide's law. André Gide, who won the Nobel Prize for Literature, said that everything that needs to be said has already been said, but since no one was listening, everything must be said again. This came from Autumn Leaves. There are a number of aphorisms in the front of Autumn Leaves that I ran across this gem there. So Rudner can be excused for not having read what Pascal had to say about this. Pascal discovered it 300 or 400 years before Rudner did. So, you know, you've heard about Schrodinger's cat. Well, we're going to talk about Pascal's duck here, making decisions using contested knowledge. And Pascal's ideas are really excellent here. So, by the way, it only took me a minute to find, I Googled duck with a beret. It took me a minute to find this cute little guy. And Pascal's wager is what we're going to look at. It comes from the Pensée. This is the version I use, which also has disturbingly perceptive psychological penetration and extraordinary intellectual insights into the human condition. And that's why I find it useful in addition to his wager, which we're going to talk about now. Pascal's wager, you probably heard about, is usually handled in a 2x2 decision table. God exists and you believe it. This is in Pascal's day, what they thought would happen. God exists and you believe it, you get eternal bliss. God doesn't exist and you believe it, no big deal. God doesn't exist and you don't believe, well, you got it right, so it's moot. And the one in red is the one that Pascal said you needed to avoid, and that was making a type 2 error, because in his day, pretty much everybody believed, not many people believe in hell these days. I looked it up, but in his day, this is what they thought you were headed for. Not a very pleasant prospect. So using Pascal's wager, eternity in hell would obviously be pretty much a 10. And even if it was only possible that God existed, Pascal said you should probably take measures to avoid an eternity in hell. But this is the step 3 I was talking about, and that is he shifted the line because he said it wasn't that tough to avoid an eternity in hell, and so he shifted the line because it was a fairly easy prospect. And so then after he shifted that line, he said, even if it's very unlikely that God exists, you still ought to take the measures to avoid this eternity in hell. So this line shift is what we also need to do with regard to the 2D6 inhibitors and tamoxifen. And when we do that shift, we find that even if it's unlikely, it clearly falls into the severity. It clearly falls into the action needed category. And how easy is it to avoid? Well, if we gave this guy a bunch of darts and we had him throw it at the dartboard with all the SSRIs and SNRIs on it, all he'd have to do is avoid these, which he probably would. Now, sertraline, maybe that's kind of a middle ground. It has some effect on 2D6. So maybe we should avoid sertraline, but certainly those three at the top. And this has, and we're getting close to the end here. This has, or reminds me a lot of the problem over the last almost half a century of the interaction between antibiotics and the risk of unintended pregnancies in women on oral contraceptives. It's a very similar issue and some of the similar problems that we run across. And just to give you a very quick overview of this, in 1975, coming out of the UK, 3 women on oral contraceptives became pregnant after ampicillin. For the next 25 years, hundreds of cases of unintended pregnancy were reported, but these are very hard to evaluate whether it was causal or not in people who took antibiotics. And there were also a number of observational studies, some of them showing a DDI and some of them not, but there were major flaws in many of the negative studies, especially. And here's where things turn bad, as you'll see in a moment. The reason why I say they turned bad, because around the turn of the century, the tide shifted and the flawed negative studies became accepted as fact. And this is what I'm afraid is going to happen with tamoxifen. The flawed negative studies are going to become accepted as fact. People will start to ignore 2D6 inhibitors, and women will have breast cancer recurrences and possibly even die as a result. So as a result of this tide shift, during the last 10 or 12 years, most regulatory agencies removed the warnings. WHO, our own CDC here in the US, the UK Royal College of OBGYN, labeling for US oral contraceptives removed it. It still occurs in some other countries. Last time I checked, Canada still had a warning. But the question is this warranted, that this idea that this interaction is not worth worrying about? Well, the flaws in the study are major. The list of antibiotics studied varied. Some combined all antibiotics together. Some included 3A4 inhibitors, which would be expected to increase serum concentrations of hormones, not decrease them. Antibiotics differ in their antibacterial effects. And so the effect on gut flora, which is the supposed mechanism for this, purported mechanism, would be different for different antibiotics. And many of the pharmacokinetic studies were way too small to detect an interaction. So the flaws were major in that study. And the landscape changed in 2020 because a study came out of the UK, the most definitive study ever published on this, where they looked at control drugs in green, antibiotics in orange, and enzyme inducers, which is not a controversial interaction. Most people agree that enzyme inducers do impair oral contraceptive efficacy. So this certainly suggested that, in fact, the interaction is real, but perhaps even more important is this e-supplement, which I have a red arrow pointing at, where this group and the e-supplement to this paper went through virtually every study that has ever been published on this interaction and critiqued it. And reading that e-supplement will convince you that that claiming this interaction is bogus is simply not rational. So we still have naysayers. We still have some believers. I would suggest the reality, again, puts it in the possible category. And if it's possible, action is probably needed. Now, in this case, and I've given 2 versions of this. In this case, suppose there's a woman who really, really doesn't want to become pregnant. For her, the severity might be up there. For another woman, it might be different. So I think that the woman should be the one making the decision on this. Tell them that there's a possibility that the antibiotic might reduce efficacy of their oral contraceptive, and let them decide if they want to take extra precautions for a few weeks during the antibiotic therapy. Condoms, abstinence also works, perhaps not as popular. But I think it should be a decision for the woman involved, because it may be very important to them, or it may not be as nearly as important. Maybe they're planning to go off the oral contraceptive in a month or two anyway and become pregnant. So this is a decision, I think, for the woman. But the parallels are there with regard to the interaction with, and we're right on time here because I'm almost done. The parallels are there between this issue of the oral contraceptives and antibiotics to the tamoxifen 2D6 drug interactions. We stand the risk of prematurely disregarding the interactions when in fact it may turn out to be real in the long run. So finally, and this one will only take a few minutes, I want to apply this drug interaction information to minimizing the risk of patient harm. So let's say we have a hypothetical patient, and I just made this up. Let's say June is 42 years old. She's premenopausal, so probably at greater risk. She's taking tamoxifen. She has been for a year. She's also on albuterol and diltiazem. June will be starting on an antidepressant. And I am making this comment that using an SSRI that inhibits 2D6 would be simply absurd. And yet many of even in the last 5 years, many of the studies looking at tamoxifen and 2D6 inhibitors have said, don't worry about it. Give any SSRI that you want to, which I think is not correct. So all you have to do is avoid those and maybe sertraline, and you've got it covered. So there's simply no reason to give her one of these that could impair 2D6. And also, I think that we need to come up with some way, and I don't know how to do this exactly, maybe a card that has all this information on it, a separate card. But we need to advise June to avoid St. John's wort for sure, other inducers, perhaps prescribed by other prescribers, 2D6 inhibitors, including OTC like Benadryl, diphenhydramine. It's not a terribly potent 2D6 inhibitor, but why give it if it could interact with, again, it's why take a chance? And then give her a list, if possible, with all these medications on the list, and have her call her health provider if anybody is planning to give her any of these drugs. And the last one I want to talk about is Maya, again, hypothetical. Let's say she's 38 years old, again, premenopausal, taking paroxetine. So she's already on the SSRI with good results. And now she will be starting on tamoxifen, 20 milligrams a day. Should she switch to an SSRI that does not inhibit 2D6? This is a little tougher. The other one was a slam dunk, in my opinion, because now you'd have to get her off a drug that's working, paroxetine, and get her on one of the other SSRIs or SNRIs that doesn't affect 2D6, with a little more of a hassle. And some people make a big deal about this, but as far as I'm concerned, the hassle of getting her switched from one SSRI to another would be a lot less hassle than having her breast cancer return or a metastasis or even a fatality. So I think that probably in most cases, it would make sense to go through the hassle of switching her SSRI. And again, as with June, we need to advise Maya to avoid all of these other drugs if possible. Okay, final slide. So we need to go back to Pascal's duck and use this 3-step process. So far, people have just been using largely the 1-step process of looking at the probability, but we need to add severity and the ease of avoidance. That's what Pascal would recommend to us, or his duck would recommend to us. Okay, so I think I just about made it exactly on time, 45 minutes, and that leaves us time for questions or comments.
So like I promised at the beginning of the presentation, this is great, Phil. Thank you very much. You're certainly a leader in the field and have thought through lots of these issues. There is one question that I'll put out there. I don't know the answer to it. and that is is the FDA doing anything currently that you're aware of about maintaining a list of both the inhibitors and inducers, an authoritative list of this, or what's the status of that? As far as
Yes, yeah, they do have lists of pretty much all of these things, and the lists are pretty good. The one problem I have with the FDA lists is that they love putting these things in categories. They say this one is a strong inhibitor of, say, 3A4. This one's a moderate inhibitor. This is a weak inhibitor. And the problem with those kinds of designations, even though the drugs they have as inhibitors in all three categories are pretty good, the problem with that is that there's so much overlap. So if you have patient A on a moderate inhibitor, they could have a much larger effect from a particular drug interaction than somebody on a strong inhibitor. So there's overlap. And so if you put too much stock in, well, this one's strong, this one's moderate, this one's weak. You put too much stock in that, you could be misled in any given individual. So, but yes, they do have lists, and the lists are pretty good.
Great, thanks for that. That's the only question I've seen at the moment. For those of you that are in attendance, if you have other questions, feel free to put them in the Q&A, and we'll try to address those. One of the things that I really liked about this presentation, Phil, was this kind of starting to consider all of the issues around a particular drug interaction. And as humans, we like to simplify things. We like to simplify it into kind of a yes/no response, but really it becomes much more nuanced depending upon the clinical situation. And as a former pharmacist, I'm no longer a pharmacist. I've given that license up. But as pharmacists that are on the call with us today and maybe listening to this in the future, any piece of advice that you want to perhaps give to them about how to quickly be able to sort this out as you're working through kind of these decision points that you might be faced with given a clinical scenario. You've got a patient in front of you, seeing a warning on the computer, don't do this, and yet in the back of your mind that that this might may be a really important interaction or may not be an important interaction. How to kind of attempt to integrate this into a clinical care scenario. So any nuggets of advice that you want to give our potential pharmacists about this?
Well, yeah, I think that unfortunately, most of the time what we get are just warnings that there's a drug interaction. And the two things that tend to be missing from that are, number one, and I know that this is something that you and I and our group, Dan, has been working on a lot, are what are the risk factors for this particular interaction? And for some, like clarithromycin and colchicine, you don't care about risk factors. It doesn't really matter. But for others, for example, one of the ones that we have worked on with our group, for example, thiazide diuretics and SSRIs being given together, causing hyponatremia, there are a whole host of predisposing factors, so that one patient A might be at very low risk from this interaction, and patient B might be at very high risk. I won't go into the details, but so risk factors are one thing. So to the extent that you can, and I realize it's difficult to do on the fly, but try to apply risk factors to that interaction and say, is this particular patient going to be at risk from this? And the second part of that then is management. And this is the part that frequently is left out. What should we do about this interaction? What are good ways of solving it? And with the SSRIs and tamoxifen, management is very easy. Use one that doesn't interact. So in that case, it's a very simple management issue. So focusing on risk factors and management, I think, are the two things that we have to give. And pharmacists need more information given. They can't keep all that up here. There's just simply no way. I long ago gave up on trying to remember every drug interaction. And it's not just age, Dan. But but nobody can remember it all. So we need to provide pharmacists with more of this risk factor and management information, not just there's a drug interaction, but what are the risk factors and how do we manage it?
Great. Thanks, Phil. Yeah. We did get another question in that I'll put out here. And what do you feel about electronic health record decision support tools surrounding drug interactions and genetic lab results? So, could we give that information to prescribers when ordering tamoxifen? So I'll let you give your take, and then I'll chime in on that question as well, if you don't mind.
Well, yeah, and obviously, since there are so many with tamoxifen, there are so many factors that can affect this, both from a genomic standpoint and from an other drug standpoint. That all of that stuff, it would be ideal if all of that stuff could be considered all at once. And if you had all of that information, if you knew the status of this patient, are they a high, medium, or low as far as activity of ABCB1 or those kind, if you had all that information, you really could identify people who were at higher or lower risk, and it would be very useful to have. So yeah, I'm very much in favor of that. Of course, we're in the process, we're headed towards that direction, I think. So Dan?
Yeah, well, I was just going to say, Phil, yeah, that's kind of the holy grail of how we would incorporate, this information together, the pharmacogenomic information at the patient level, the risk factors, obviously, along with the drug characteristics. And I know that, many of the organizations that provide information about drug interactions are starting to embed into their systems, the genetic information about metabolism, what have you. Although trying to actually make that actionable at the prescribing decision, I think is we're just not there yet. We're shooting for the moon, and we're still building the spaceship on the ground. So I think we're quite a ways away from actually being able to make that happen. Although I do know, people are thinking about it, working on it, but I don't know of anybody that has been successful at putting those together yet. So, but if there's anybody on the call that wants to chime in on that, feel free, let us know, and we can unmute you and you can describe your process. But yeah, we hopefully will get there. Kind of keeping an eye on the chat. I don't see any other questions at the moment. So I'm not going to, I guess, keep you any longer if there's no additional questions, but I do want to thank our presenter, Dr. Hanston, again. Thank you very much. Yes, he has been doing this a long time, probably not as long as I had indicated earlier on, but it relies on some assumptions on his behalf and my behalf. But nonetheless, Phil is really well known and just a true scholar in this field. So thanks for bringing it, both the issue of drug interaction with respect to tamoxifen, but also the philosophical components of how we might want to approach drug interactions, not just this drug interaction, but all drug interactions. Right. How do we try to put this all together to keep our patients safe? So with that, we'll go ahead and stop the recording. We'll make this available on our website. And our website is, again, and I'll put this into, I guess I try to put this into the chat here on the fly, but you can visit our website at ddicds.org. You'll be able to access this recording and other recordings that we've done for webinars. So thank you very much for your attendance today. We wish you the best. Take care.